Our next speaker is going to be Liji Wang, who's at Iowa State University. He's actually in the engineering department. So this will be this will be an exciting talk, and perfect. All right, I'll hand it over to you, Liji. Thank you.
Is there a pointer?
Pointer is right here, the green.
Green.
And then do you want a lapel mic or do you want this mic?
Thank you. Okay, thank you for the invitation, for the introduction. Good morning, everybody. My name is Liji Wang. I am an engineer, and to be specific, I'm a industrial and systems engineer. I work in the area of operations research and data analytics. So for more than a decade, I have been working very closely with plant breeders and plant scientists to design novel algorithms to improve the efficiency and effectiveness of plant breeding. So today, my agenda is to introduce my work in three major categories of models in operations research and data analytics. The first category of model is called descriptive model, and they are trying to explain how a complex system works and why it works the way it does. I will use G/E interactions as an example. And second category is the predictive model. It's trying to predict the output of a complex system, especially for new and unseen input conditions, and I will be using a breeding simulator for that example. The third category is prescriptive model, and that's trying to optimize the intervention that we can impose to the system to improve the output from the complex system. And for that, I will be using genomic selection as an example. So let's start with the descriptive model for G/E interactions. So you know there are so many models proposed for G/E interactions, and we have seen quite a few today in the presentations. But more recently, we have been trying this new model we call this data-driven crop model, which is basically a hybrid of the crop modeling approach and the data-driven machine learning approach. And these are the three diagrams showing you the differences between the three approaches. So crop model, as you know, is a mechanistic model describing the growing process of the crops. So it integrates the best human knowledge about plant biology, plant physiology. It's very scientific, it's very explainable and descriptive, but it does suffer two limitations. One is it has a lot of traits, maybe dozens of traits, that need to be experimentally calibrated. So I remember a couple of years ago, we were working on this NSF project, and we had to estimate the parameter of leaf appearance rate. So we sent students to count the number of leaves every day. That was a very time-consuming, labor-intensive work, and that's only one parameter. So talk about dozens of parameters, very labor-intensive. And second limitation is that even if you calibrate all the parameters, the predictive performance of crop models typically are not as good as some other models. And then we are also looking at the data-driven machine learning models, which are very powerful in terms of the predictive performance. But on the other hand, it also suffers two limitations. One is it's very sensitive to the quantity and quality of data. So if there's not a lot of data, if there is some bias in the data, the performance may not be that good. And also, it's typically a black box model, so it cannot be very explainable or descriptive. So what we are trying to do is to combine these two models and create something called the data-driven crop model. So in the core model is a crop model, so it's scientific, it incorporates human knowledge, but it doesn't need to be the parameters do not need to be experimentally calibrated. Instead, we are using the machine learning model and historical model to calibrate the parameters so that this model has very good predictive performance and also completely explainable and descriptive. So let me give you an example. This is an example that we recently created. It has hundreds of equations, but this is the core modules of the model there is the soil water nutrient dynamics module, there is the water nutrient uptake module, phenology clock, radiation reception, and daily biomass accumulation, and metabolism and stress accumulation, and into different organs, different growing stages. I want to draw your attention to this G parameter. So the G parameter represents the set of genotypic parameters that are fundamentally determined by genotype, not determined by the environment. Because this is very useful, very useful especially for breeding purposes, because you may have a new variety that gives you really good yield, but you are not sure if the high yield is because of genetic superiority or is it because of better environment. And so let me give you some examples of the G parameters. So in this case, we are looking at 20 different clusters or 20 different varieties. And these are just 3 examples of the genotypic parameters. The top one is the heat stress temperature. That's if the temperature is above this level, the heat stress starts to kick in. So we can tell that the cluster or variety number 11 has the highest heat stress tolerance, but number 19 has the lowest heat stress tolerance. And this is the drought stress tolerance. So the water level goes under this level, the drought stress starts to kick in. And the number 7 or 8 has the best drought tolerance. And this is the best temperature for photosynthesis. And so what can we do with this set of parameters? Let me show you a case study we recently finished using the data we collected from NAS. So this is the county level corn data in the past about 4 decades. The vertical is for the genotype. So we are making the assumption that, let's say, every year we calibrate or we calibrate one average genotypic parameter to represent the average genetic performance in that year. And these are the 40 years of genotype. So you can see the evolution of the genetic performance over the years. And the horizontal is the environmental conditions, including weather and soil and management practices over the year. The diagonal is what we have observed historically. We grew, for example, the 1981 seed in the 1981 environment. We grew 2020 seed in the 2020 environment. But this model allows us to answer the what-if question. What if we grew those seeds in all those years and environments? What's going to happen? So you can tell that over the years, the color map is, this pink color is 11 tons per hectare. And at the bottom color, that's 4 tons per hectare. So you can tell over the years, on average, the genetic performance is quite dramatic. It's quite noticeable. But on the environmental side, there is not a clear trend of improvement or the other way. But you do see some very noticeable years, such as the 1983 drought, 88 drought, 93 flood year, and 2012. You can see 2012 was so bad that even the recent seeds, recent genotypes would not have done too much better than the 2012 seed, which was actually not that bad. So the 2012 drought, a lower yield, was mostly because of weather, not because of genotype. So that was the first type of model. And second category is the predictive model. So we know that plant breeding is a very slow game. And if you make a decision, it takes you maybe half a year or even a year to get some feedback how good your decision was. So what we are trying to do is to provide you some breeding simulator so that you can ask the what-if question very quickly. So that breeding simulator will give you feedback, will predict what's going to happen as a consequence of your breeding decision. But in order to make a good breeding simulator, I really want to clarify 3 definitions. These are what's nature, optimizer, and what's a simulator. So what's an optimizer? I define an optimizer as a decision maker. So if a breeder is making all the decisions, the breeder is the optimizer. So the decisions are include, so you take the genotype data, phenotype data, and then you do some genomic prediction, genomic selection, and then you determine the crosses to be made. So that's a optimizer. If you are using a software decision support tool, so that's also part of the optimizer. And then what's nature? Nature does two things. So after the crosses are made, it's going to determine the genotype and phenotype of the new generation of the varieties. So the genotype is determined through the meiosis process, and the phenotype determined by the G/E interactions. So this is how the optimizer and nature interacts. Every year or every generation, taking the genotype data, phenotype data, the optimizer will determine the crosses to be made, and nature will give you the new genotype and phenotype in the next generation. It comes back again and again. But suppose you have a new algorithm for optimizer. We are not going to test it in the real breeding program because it's very risky, very time-consuming, very expensive. Instead, we are building a computer simulation to test it. So we are building the simulator in place of nature. So if you look at the literature, people are focusing a lot on optimizing the performance of the algorithms, but very little attention is paying to the design of the simulator. But let me point out the danger of not paying attention to a good simulator. So the biggest difference between nature and simulator is that we know very little about nature. So through thousands of years of human civilization, we have learned a lot about nature, but compared with the whole set of knowledge there is for us to learn, how much have we learned? I would say much less than 1%. But what about the simulator? When people are designing a simulator, people assume that we know almost everything about the simulator. So that's very dangerous because you may be designing an optimizer that's playing the game, that's working very well in the simulation, but when you plug it into the real breeding program, it will definitely disappoint. So my point is that when you are designing or when we are designing a simulator, we have to make sure to make it opaque, not make it transparent. So the transparent is what we have seen in the literature, and the opaque simulator is what we are proposing. So in terms of the genotype, a lot of the simulators are assuming that if we are using 1,000 markers, that's all there is in the whole genome. But I believe that we should be designing an opaque simulator so that even if you or the optimizer can only see 1,000 markers, there may be still millions of genes there is in nature, and that should be integrated in the design of the opaque simulator. In terms of the allele effect, some of the studies are assuming the allele effect, the true allele effect is known by the optimizer. It's the same effect used by simulator and optimizer, but that's not true. Nature doesn't tell us the additive effect, non-additive effect. Just because you are assuming there is no non-additive effect doesn't mean there is no non-additive effect. There has to be some dominance epistatic effect in the opaque simulator, and also many people use the linear equations for the phenotype, but I think we should be using a more complex process-based mechanistic model for the G/E modeling. Okay, just for the sake of time, I will move on to the third category of model, this prescriptive model. I will use genomic selection as example. So first I want to point out one misconception about genomic selection. So many people equate genomic selection with genomic prediction, followed by a straightforward truncation selection. So over the years, we have been trying to prove that genomic selection should be more than just prediction plus truncation selection. Actually, we can do more strategic selection than truncation selection. If you do that, you will improve not only the genetic gain, but also genetic diversity. So here we are comparing a set of 5 different genomic selection algorithms in terms of different criteria. So this is very classical GBV-based genomic selection. It's a truncation selection. The horizon is G0, means we are selecting the best individuals in the current generation. And then in 2015, there was the optimal haploid value selection proposed. It's also a truncation selection. but the horizon is 1 generation later. So we are selecting not the best individual in the current generation, but the best progeny that could be produced in the next generation. And then in 2017, our group proposed the optimal population value selection algorithm. So it's no longer a truncation selection. So it's based on the group selection concept. I will explain with an example later, but also looking at the G, and that's the you are selecting for the best individual in the future, not in the current generation. And then 2019, we proposed the look ahead selection. Not only it's a group selection looking at the future, but also we're optimizing the mating decision and time management. So your optimal selection may depend on your selection timeline. And recently we published a paper for optimal resource allocation. I will give you some examples. So this is the GBV selection. You are estimating the GBV, you rank them, and then you cut a line for truncation selection. And then the OHV is saying, okay, let's consider this example. The yellow alleles are favorable, blue alleles are unfavorable. These two individuals, which one do you select? So GBV is counting the number of favorable alleles, is selecting the left one. OHV is saying, let's select the right one because in the next generation, so this individual may give you a perfectly favorable gamete in the next generation. And then the optimal population value selection approach is saying we should be looking at the selection as a group, not as individuals, because let's say we are selecting 2 out of the 3. Which 2 are you going to select? The GBV and OPV all select the first 2 because they have much more favorable alleles than the third one, but the OPV is saying look at the complementarity in the first one and the second one. So maybe in the future generation, their progeny will be able to have all favorable alleles, but if you select the first 2, the last locus is fixed with an unfavorable allele. And then the look ahead selection is saying when we are making selection, we should be looking at the future. So it's like playing a game, playing a chess. You don't just look at the first move, you have to be reading multiple moves down the road, then you will be very strong player. So similarly, when you are making a selection, you have to look at in the future. So you are maximizing the performance of a future generation by optimizing your selection in the current generation. So if you do this, you will be getting much accelerated genetic gain as well as much higher genetic diversity. And finally, this is the resource allocation. I will show you what I mean by resource allocation. So the even allocation, this is the typical approach, is we are spending the same amount of resources, we do the same number of crosses, producing the same number of progeny every generation. But the dynamic approach is saying we may be allocating our resources more strategically depending on the outcome, depending on, for example, the recombination frequency and a lot of other uncertain factors. If you do that, you spend the same total amount of resources, but you could be getting much better outcome. And let me show you a case study. So this is a case study using some maze data. The horizontal is genetic gain, vertical is the percentile. So what we did was we did thousands of times of simulation, and this is the best, this is the worst case, that's the best case. So this is actually the cumulative distribution function. The better approach will give you a curve that's further to the right. So this is a GBV, this is OHV, is better, and this is OPV, a lot better. This is look ahead, somewhat better, and this is resource awareness look ahead, so much better. So I hope I have convinced you that we can do much better than truncation selection if we are more strategic. So finally, I want to thank my collaborators and supporters, especially give a special shout out to 3 centers on campus. I was the campus Plant Sciences Institute, AI Institute for Resilient Agriculture. That's a $20 million new grant funded by NSF and USDA. And also this Track Translational AI Center. So these 2 centers are looking for industry partners and academic collaborators. So if you're interested, feel free to reach out. With that, I will stop here and thank you for your attention.